would you feel if you received a drug that caused you problems? How would you feel if you later learned that the reason that those drugs caused problems might have been because the animal data on which they were based was sloppy or even fraudulent? Now, this is a recurring nightmare for regulatory agencies charged with assuring the safety of medical products. The main tool that we have to assess the safety of a drug before it goes into clinical trials is our animal data. So if that data is wrong, we have a very scary situation. Now, this is the situation that presented itself in the 1970s. At that time, regulatory agencies demanded that drug companies show them animal data before they conducted their clinical trials. As part of their application, they had to produce a set of animal data that would prove the safety, at least, of the product. However, what was missing at that time were any rules by which that testing had to be done. As a result, animal testing became a huge and unregulated business. One of the biggest companies in this business was a company called Industrial Biotest Laboratories. Now, Industrial Biotest Laboratories was huge. It did about 40% of the toxicology testing in the US. However, they conducted their activities very unprofessionally. If you went to their laboratories, you might find that their personnel were not trained. You might find that animals that died remained undocumented, that cages had loose doors so that the animals could get out at night and socialize. As a result, most of the trials that Industrial Biotest Laboratories did were found to be invalid. 4 managers of this company were later jailed for fraud. However, the Congress and the public were outraged when they found out about these kinds of problems. They demanded that rules be written to prevent such kinds of fraudulent and sloppy behavior in the future. Now, this probably comes as cold comfort to the people who received those drugs that were untested. However, FDA promptly went to work and developed a set of rules that was called Good Laboratory Practices. Good laboratory practices are a framework. They're a set of rules designed to ensure the quality and integrity of the animal data you collect. Now, good laboratory practices don't govern all kinds of experiments. They are primarily written to govern a certain type of animal testing, not bench testing, but animal testing, that's going to be used to support applications to commercialize medical products and applications that go to the FDA. Thus, they are seldom used to do experiments in a research laboratory in a university. Good laboratory practices can be very complicated to implement. Thus, even large companies often outsource their animal trials. There are specialized contract research organizations that do nothing else but those kinds of specialized animal trials under good laboratory practices. On the other hand, some companies prefer to keep their animal testing in-house. This may keep the costs down, and it also may ensure that they have tighter control over the way that the testing is done and the documentation is maintained. Now, you may be watching this video because you're trying to decide about whether you're going to outsource your testing or do that testing yourself. Now, this decision is complex. It depends a lot on the resources that you have, but it also depends on how many tests you're going to be doing, whether or not you're going to be wanting to maintain a full-time staff year-round in order to conduct this testing. What we hope you'll get out of the video today is some ideas about what would be involved in setting up such testing in order to guide the decisions that you make. GLP regulations can be found on the FDA website. They're for free. All you have to do is click on the homepage, www.fda.gov, And on that page, you'll see a link to the Code of Federal Regulations. When you open this site, it will show you the whole listing of all of the different regulations in the 21st title. If you just scroll down, you'll find the GLP regulations in Part 58. Now, the GLP regulation document is not long. However, you're going to have to read it if you want to understand the extent and nature of the regulation that you'll have to satisfy if you're going to run a GLP lab. The GLP regulation as a document is divided into several sections, and each of the sections deals with a different aspect of laboratory activity. At the very beginning of the regulation, you will find a number of definitions. Now, if you have a science background, you may not want to go over all of these definitions here. Some of them are pretty obvious, but perhaps we should refresh you on just a couple of the more specialized terms. What, for example, is a test system? You have three choices. Is it an animal, is it a piece of equipment, or is it a laboratory? The correct answer is animal. For example, if you're doing a toxicology test in a rat, the rat is your test system. In association with the test system is a test article. A test article is the product that you are testing, the drug or the device whose safety you're trying to evaluate for the purpose of your FDA application. In addition, you'll see the term control article. I think it's pretty obvious perhaps now to you that the control article would be the placebo or the active control, perhaps a positive control, that you know will evoke a consistent response. And this is a comparator for your active test article. There are also definitions associated with the organizations that are doing the testing. You will see the term testing facility. This term is used for the site at which the animal testing is done. If the animal testing is outsourced, this would be the contract research organization that is doing the testing. In contrast, the term sponsor is reserved for the organization that is paying for the study. If the trial is outsourced, then the sponsor would be the company that is paying the contract research organization to do the animal testing. If the sponsor, on the other hand, is doing the testing in-house itself, then the sponsor and testing facility may be one and the same. A very important early item in this list of things over which the FDA requires that you have control is the training of your personnel. The FDA wants to be sure that the personnel that work for your organization know how to do the jobs that they are given. Thus, they require in the regulations that the personnel have the training and experience or combination thereof that will show that they are able to do the jobs that are required in your experiments. Now, how are you going to show the FDA that your personnel are appropriately trained or experienced when they come to do an audit? Now, typically, this is first done by having a job description for each member of your staff. The job description will frame the kinds of training that would seem to be required for that job, wouldn't they? Then you're going to have to maintain training records that show that the person has acquired the training or experience to carry out the job description. You will see the term study director. This is the person who is going to take primary responsibility for the conduct of the study at the testing facility. Both the study director and the quality assurance manager need to understand the degree to which such studies are going to involve paperwork. The quality assurance manager will need to maintain records of protocols, master records of studies that are being done at the facility, and records of audits that are conducted of each study under his or her control. They also will have to have a list of dates the audits were done, dates that reports were given to management, and these can be presented to the FDA when the FDA audits. There must be adequate paperwork to show that all of the people involved in the study did their jobs appropriately. Each animal in the study may end up with a medical file that is much like the medical file that you would have if you were in hospital. Both the study director and the quality assurance manager need to understand that paperwork will be a continuing burden. The paper that is generated by the quality assurance unit will contain not only copies of all of the protocols of the studies being done in the facility and a master log of all those studies, but it needs to contain all of the audit records from all of the visits that were done to assure that the study is in compliance with the regulations. When the FDA visits, the auditor will be looking for a list of specific dates for the audits and dates at which the audit results were reported to management. The principal investigator, similarly, will have very large files that contain copies of the protocols under his or her direction, copies of standard operating procedures and methodologies that they are using, certifications of lab reports, permissions of animal ethics boards. Each animal, in turn, will end up having a sheaf of documents that will look very much like the medical file that you gather when you go to the hospital. The FDA will be looking at all those documents because documentation is about the only way that, in retrospect, they can assure the quality and integrity of the data in the trials that you did. Facilities can be particularly problematic for especially a small company that is considering whether to set up a GLP facility for the first time. Now, the FDA has some specific language. What it says is that each testing facility shall be of suitable size and construction to facilitate the proper conduct of non-clinical laboratory studies. It shall be designed so that there is a degree of separation that will prevent any function or activity from having an adverse effect on the study. Now, these seemingly simple words have many implications. Remember that there are a number of areas in any animal facility. There are areas where animals are held, where their cages are located. There are areas where the cages are cleaned and sanitized. There are areas where we store test and control articles, areas where we store food and water, areas where we test food and water. Remember that the food and water that are used in these studies has to be analyzed and assured to be sure that the animals don't get sick on it. If any one of these areas is compromised, it could have a negative effect on the outcome of the trial. A GLP facility has to be particularly careful about ensuring an adequate size, design, and construction. Now, some of the aspects that we have to consider here are whether or not the facility can be cleaned properly. We have to be concerned about its temperature and its humidity. And we also have to be concerned about light-dark cycles because many animals are affected by those cycles. In each case, we have to have a way of monitoring the activities relating to that management and documenting that appropriate actions were taken. As a result, putting together a new laboratory facility can be a multi-million-dollar operation. If you're a small company and you do not plan on doing large numbers of trials, this requirement may be very expensive and may cause you to rethink whether you yourself want to do the trials in-house. If that company is only planning a few trials, intermittent trials, the multi-million-dollar costs of setting up a proper facility may be daunting and may be a reason why they should investigate the use of a good laboratory facility in a contract research organization. Facilities operations can be very challenging as well. You can have a beautiful facility, but if you don't manage it in an appropriate way, it will be all for naught. The FDA will expect to see a large number of standard operating procedures that are designed to govern the activities that occur in these areas. These may include things like how you're going to ensure temperature and humidity control, how you're going to monitor temperature and humidity, where you're going to buy your animal food, and how you're going to analyze that food to ensure that it meets its specifications. They're going to be concerned about things like your water. Are you analyzing your water? Where is your water coming from? There will need to be procedures written that govern the identification of animals, the necropsy of animals, and other animal-related procedures. Here is a list of at least some of the standard operating procedures that will be expected in your organization. Thus, you're going to have to be sure that you set aside enough time to be able to write these standard operating procedures and to have them reviewed and approved for use in your facility. Standard operating procedures are wonderful to have. They're a great training tool, and they allow your laboratory to function in a much more consistent way. However, there are a lot of requirements that govern how you manage those standard operating procedures. For example, if you change a procedure, you will need to change your standard operating procedure documentation. You don't just do this on the fly. You must make sure that those changes are reviewed and approved, and you should archive all the former standard operating procedures, so when the FDA audits, they can see that you are changing your operating procedures in a controlled way, and they can see evidence of what the previous procedures were. So this adds to your archiving file. Now, standard operating procedures will take a lot of time to write, so you should set aside enough time to ensure that these can be put in place before you start your experiments. Another area of focus in GLP regulations is the calibration and maintenance of equipment. There are many pieces of equipment in a laboratory. The ones of particular importance are those that will in some way affect the potential validity of the data collected in the animal experiment. For those pieces of equipment, it's very important that there be standard operating procedures regarding calibration and regarding maintenance. As a result, each of those pieces of equipment will end up with its own little file. In that file, you may have its calibration SOP, its standard operating procedure, its maintenance procedure. You'll probably have a log that shows calibration intervals and is signed off each time calibration occurs. Similarly, maintenance logs need to be kept so that you know when the machine was maintained, and the file will have reports, perhaps reports on situations in which the equipment failed and had to be maintained, or any other reports that relate to its function. When you have this documentation, it is important that you keep it current. There's only one thing worse than not having standard operating procedures and documentation, and that's not following the documentation and procedures that you have. Each time a study is conducted under good laboratory practice conditions, it is important to have a protocol. Now, a protocol is a very detailed, step-by-step tree or description of how the experiment is going to be conducted. And the experiment cannot start until that protocol is signed off by the study director. Now, when the study director signs that report, he or she is essentially signing a legal document, a contract. They're trying to ensure that you stick to that protocol. Thus, if you do not follow every step of the protocol, you will have to write a note to your file, what we call a protocol deviation, to explain why you deviated from the protocol. Now, this is where these kinds of studies look very different from studies that, say, are done in a university laboratory for basic discovery. In such studies, GLP conditions might constrain the nature of creativity or innovation during the study. It's not unusual in a typical research study for the protocol to change from one experiment to another. However, in safety studies that are going to be presented to the FDA, it's very important that the protocol be conducted each time in a standardized way. In fact, many of the protocols that we used are actually referenced from standards organization, like the International Standards Organization, where you can find specific protocols for certain types of animal testing. Now, when you're writing such a protocol, there are several things that must be kept in mind. Usually the first page of the protocol is a stylized cover sheet that includes a lot of information. Information including the number of the study, the title of the study facility. You can find the listing of things that should be typically on such a procedure elsewhere. When you are writing the protocol, you must be very careful to think about every step in the protocol that needs to be documented. Remember, if it's not in the protocol, you cannot carry out that step. So you have to walk step by step through all of the procedures from the beginning of purchase of the animals to the end, where the animal data has been taken, where the animals have been necropsied, and where the tissue has been analyzed. Your protocol is your roadmap for the study. Now, as you do each animal in the study, you will want to document very carefully what happened at each step. Now, typically, some of that data will be collected on specialized forms, just like case report forms in clinical studies. That will allow you at the end of the study to pull all the data together easily. Once all of the documentation is collected for each animal, it's drawn together into the animal file. So the animal file will not only have perhaps data points that were collected during the experiment, they may have records that come from blood analysis from other laboratories, they may have nursing notes, anesthesia records. Everything about that animal should be collected together so that later when the study is analyzed, you can go back to those data at any time. Those are data that will probably be pulled during an audit by the FDA to assure that the data that appears in your final report that's written at the end of the study correlates with the data you collected during the study. The study ends on the day that the study director signs off the final report. Records from animal studies are precious, and they must be archived after you finish the trial. Now, archiving requirements can be for very long periods of time. It's not uncommon that the data must be kept for at least 2 years after the approval of a marketing application for which that data has been submitted. Now, if you have multiple applications over time for related products, that can mean that this data needs to be kept for even decades. So you have to think hard about where and how you're going to retain your data. Now, FDA typically requires that samples be retained from the animals on which the experiments were done. This can include, for example, keeping body parts or even whole carcasses in a way that they can be audited at a later date. So you will have to think out very carefully how you're going to deal with this so that over time they do not get lost or damaged, for example, by fire, flood, or a move. If you're working with a contract lab, you have to arrange that the material from that contract lab is either archived at the contract material or comes back to you in a way that you can keep it for a long time. You do not want to lose that data because audits often occur several years after the data was collected. In addition, sometimes that animal data is used to support multiple commercializations. So for such applications, you'll need to hold that data for a very, very long time, often a decade or more. So at this point, you're probably saying, these GLP requirements look pretty tough. Is this really a road I should be going down? Now, in some situations, the answer to this question is clearly yes. You may have experiments that have specialized protocols that are going to be difficult to do in a contract research organization. Or you may find that you need a situation in which it's very convenient for your personnel to work in the animal lab without flying often across time zones to find a lab that can work with you. However, if your experiments are more straightforward or you're not doing them very often, you may want to consider the use of a contract testing laboratory. Contract research organizations are in the business of doing animal trials, and they can be very knowledgeable as well as very well equipped for some of these trials. On the other hand, you have to remember the lessons that we learned from industrial biotest. Contract research organizations are only as strong as their staff. Sometimes the staff that they hire may not be invested in the results in the same way that you are. If they're sloppy or disinterested, that can translate into poor results in the study. So it's important, even if you are outsourcing the data, to realize that you have the responsibility. Thus, it is expected by FDA that you will physically go and talk to and audit the facilities that are doing your trials. In such a relationship, your contract should clearly spell out what are their responsibilities with regard to GLP and what are your responsibilities in relationship to the direction of the protocol. I hope that this short overview has helped you to understand some of the special requirements that good laboratory practices will impose on your experiments. Now, animal studies are taken very seriously by regulatory agencies, and so that you have to be sure that you follow these regulations very well. There's nothing worse than going to all the expense and trouble of very expensive animal experiments only to have them rejected by the FDA when you do your marketing submission. This can set your project back by many months and many hundreds of thousands of dollars. That would hurt a lot. So whatever your decision about the path that you take, make sure that you plan well and consult others who followed the same path.